Hello dear learners. In our today's video, we will start on with the learning of the process of transcription, that is DNA transcription. Now first of all, we should know that what is the meaning of transcription. So transcription is the coding of DNA into RNA. According to central dogma, the DNA when converted into RNA or from the DNA a new RNA is synthesized, the process is referred to as transcription. Now the new RNA which is synthesized is generally mRNA, but it can also be tRNA, rRNA, hnRNA and other forms of RNA. Whenever the transcription has to start, we know that from DNA the template will be taken, and through this template the RNA polymerase will work and then the RNA will be formed. So now there should be the appropriate DNA present of which the transcription can be done. Now generally, out of nowhere the transcription cannot start. The transcription can only start when there is a particular region in the DNA which is called as a gene or the group of genes. So basically these group of genes together, their transcription takes place and from them a particular mRNA is formed. So if the gene is present, then only the transcription will take place. Now when I talk about gene, I talk about the full gene, that is the fully functional gene along with its promoter and terminator. So until and unless the promoter region, the coding region and the terminator region are not present inside the gene, the transcription will not be as simple. So when the promoter, coding and terminator regions are there, then the transcription will take place. We have already told that the transcription will take place of DNA. The strand of DNA will be considered as the template strand. Now the another question arises is that which will be the enzyme or which will be the main protein which is involved in the transcription. So here, since we are using DNA, so the protein or the enzyme will be dependent on DNA. So it is called as DNA dependent RNA polymerase. So the enzyme is DNA dependent RNA polymerase. So basically it is RNA polymerase, but it depends upon the DNA. It uses the DNA as a template. Now, from where will the transcription start? There is a particular site in the DNA through which the transcription process will begin. Now, this particular site inside the DNA is referred to as the start site. Suppose let us consider that the start site is here. Suppose this is my, now again, this start site can be just after where the promoter finishes. From here also the start site can be there. Here also the start site can be there. And even the start site which is covering most of the promoter can be there. So there is a specific point where the start site is present. So let us assume our start site is present over here. Now, this is a particular point which is referred to as a start site. All those nucleotides or base pairs or mers which are present to the right of these start sites, they will be categorized as the nucleotides present on downstream. So to the right of the start site is the downstream region, downstream, and to the left of the start site is the upstream region. The nucleotides which are present on the right side are categorized as +1, +2, +3. They are named like this. They are numbered like this. This is the 0, and then all of those who are present on the left of this start site are -1, -2, -3, and they are said to be located upstream. So in many of the cases, most of the promoter part is located upstream. Always remember that after the start site, all those which are present to the right, they will be acting as a template, and all those which are present to the left of the start site, they won't be acting as the template. So they won't be coded, but the elements which are present to the right of the start site will always be coded. So in most of the cases, approximately 50 to 60% of promoter is not coded. The remaining part may be coded, and then the coding region definitely has to be coded. When I say the term coded, it means that the RNA polymerase will recognize that particular sequence, will bring in the required RNTPs, will do the complementary base pairing, and hence will generate the mRNA. So this is the process of transcription and how it will initiate. I have also talked about and I have made the clarification about start site, the downstream elements and the upstream elements. Now the question arises that which strand will be synthesized or which strand will be used as the template strand. So there are two strands we know by nomenclature, and in replication also we know this through replication that there are two strands of the DNA. One of them is referred to as the template strand. For the replication process, always remember that this strand, now see, this strand is 5' to 3' and this strand is 3' to 5'. Always this lower strand, whose direction is from 3' to 5' will by default, please note my term, by default it will act as a coding strand. Sorry, not coding, it will act as a template strand. The meaning is very simple. This will serve as a template. And when this will serve as a template, the DNA dependent RNA polymerase has to run on this particular template. So the DNA dependent RNA polymerase, let's say if I'm making this as my DNA dependent RNA polymerase, so it will run on this particular strand and it will recognize the proteins, sorry, it will recognize the particular nucleotides which are there on this strand and then will make the complementary RNTPs. So always remember this serves as the template strand, while the above one, this strand will serve as the, sorry, this strand will serve as the coding strand. And why it is called as the coding strand? Because the mRNA so formed will have a coding very similar to this, except that the thymine will be replaced by the uracil. That's why it is called as the coding strand. Although it does not play any significant role in the whole of the transcription. So this is also what we have to know about them. So now if this is crystal clear to you, the next important thing which comes is that what are the other names of these two strands? So please remember that this template strand is also referred to as non-coding strand, and that makes the obvious sense. So it will be called as the non-coding strand, and along with that it can also be called as anti-sense strand. So it is either called as the non-coding strand or it is called as the anti-sense strand. And the strand above, it is coding strand or it can be non-template strand and it can be the sense strand. So I'm not writing them, these are quite obvious. So I have told you one important point which you need to know is that by default always the transcription will be done by recognizing 3' to 5' as the template strand. Now we should talk about the prokaryotic and eukaryotic transcription. There are the minute differences which you should know. Before I start, please make this clear in your heads that when I talk about this transcription, I will designate as at TC. Now when it is occurring in prokaryotes, it will be occurring in the nucleus-like region, because in prokaryotes there is no true nucleus. And when I talk about the eukaryotes, this transcription will be occurring inside the nucleus. Sorry. Moving on further. Sorry. So now we have to talk about the prokaryotic transcription. Firstly, we will talk about its promoter. Now the promoter of the prokaryotes is a very special promoter. I'll tell you why. See, basically the promoter is in this way. Uh, Let me draw it with a different color. Yes. Okay. Suppose this is my promoter. Now what happens to this promoter is that here we have the two boxes which are present in this promoter. One box suppose this and one box suppose this. Now I'll also highlight these boxes so that you understand it in a better way. I hope this highlighted color is visible to you. Okay. Now this box, first box and second box, and then here we have the start site. Now this first box from the start site, this first box is located at the distance of approximately 10 base pairs. So if this start site is designated as 0, this will be designated as -10, and on this -10 this is particularly located, and this box is referred to as Pribnow box. A box is nothing but a sequence of nucleotides, and this box is referred to as recognition box. Recognition box, and it is located at the -35 base pairs. So there is the Pribnow box and then there is the recognition box. It is rich in GC, so it is also called as sometimes GC box. And this is Pribnow box. The sequences here are TATAAT. Please do not confuse it with Tata box. The Tata box is present in eukaryotes, not in prokaryotes, and the Tata boxes have the sequence of TATAAA. We'll learn this when we talk about the eukaryotic translation. So this particular Pribnow box is not the Tata box, for your clarity. Now we have to talk about the RNA polymerase. So we have talked already about the promoter, that how the promoter will look like. I can also magnify it so that it is clearly visible. Hmm. Now let's talk about the second important aspect here. The second point is about the RNA polymerase. So here, firstly, there is only a single RNA polymerase. There are not multiple RNA or a diversity of RNA polymerase. There is a very simple RNA polymerase. This RNA polymerase can catalyze the transcription in the two cases. By default, it can catalyze the transcription in downstream. Downstream means that time it will recognize the template strand as 3' to 5', which is in most of the cases. If this RNA polymerase is catalyzing the transcription in the upstream direction, that time the template strand which it will recognize is 5' to 3'. So these are the two cases. Now the RNA polymerase is again made up of two subparts. So I will write it over here. Uh, hmm, Let me write here. This particular RNA polymerase is made up of one, there is core RNA polymerase here, core RNA polymerase, and this core RNA polymerase is made up of units like beta, beta dash, alpha, alpha, and omega units. Secondly, there is a factor which is the initiation factor, it is sigma 70 factor. So both of them, the core RNA polymerase and the sigma 70 factor together, make the RNA polymerase. So please learn this, that there is one of them is the core RNA polymerase, one of them being the core RNA polymerase, and the other of them being the sigma 70 factor. Now, how will the process initiate? So, let's move on to the process of the transcription in prokaryotes. So now, for the simplicity, I will be drawing the structures like, wait, mm, let me draw it like this. This is my one strand. I should even draw it a bit. One strand, this is my another strand, and here I have the promoter, here I have the coding region, and then here I have the terminator region. This is my promoter region, promoter region, coding region, and the terminator region. So now what happens and how does the process initiate? So for the whole process, I will draw one of the factors, that is sigma 70 factor. Suppose this is my sigma 70 factor. Suppose I am making this as my sigma 70 factor. So this sigma 70 factor is the part of RNA polymerase, and along with the core RNA polymerase, it makes the full RNA polymerase, right? So this sigma 70 factor will come and attach. Now please do not go into the details of on which strand it is coming or on which strand it is going. Please don't say that, and please don't even think about that. See, suppose if I am attaching the sigma 70 here. So the sigma 70 will basically attach itself to a place where just next to it the RNA polymerase can attach. Now we already know that there has to be a Pribno box and there has to be the recognition box. These two boxes will play an important role in bringing up of the sigma 70, in attaching the sigma 70 into that proper region. Along with that, also bringing the RNA polymerase, the core RNA polymerase. So now after this, the next step is the bringing of the core RNA polymerase. So suppose this is my core RNA polymerase like this way. Okay. Now this, after the sigma comes here, sigma 70, the core RNA polymerase is also recruited. Let's not make them like this. Let's shift them a bit so that, and also let's decrease the size of this core RNA polymerase. Yes, this is perfect. Now first of all, the sigma 70 factor will come, and after the sigma 70 factor is has arrived, now the core RNA polymerase will reach here on the promoter region. So the sigma 70 factor is a very specific and a very special feature, that this sigma 70 here, if I draw, if this was my recognition box and if this was my Pribno box, so the sigma 70 binds to both of them like this. This is my sigma 70. And then after this, when this has been done, then the RNA polymerase will come and attach onto this particular sequence. So I have drawn them and I have shown them like this way. So here, this way the RNA polymerase attaches itself, and this is my sigma factor, which we already know. So sigma factor has come, the RNA polymerase has come, and now the melting and the identification will be done by this sigma 70 factor. So first thing is that sigma 70 factor will do here two important roles. It will do the melting at the start site and the correct identification. Then it will recruit the core RNA polymerase. Now core RNA polymerase is here. Now what happens is that the core RNA polymerase has to now start what? Has to now start the process of the transcription. Now as the process of transcription initiates here, so this core RNA polymerase has to move forward, but this sigma 70 factor does not let it move forward. And it is actually, what it is doing is, it is blocking a particular site onto it. So during the initiation, the exit site of this core RNA polymerase is blocked by the sigma 70, and hence the RNA cannot move out, and it is called as abortive initiation. So whatever the new RNA is formed here, let's designate it with this color. So we already know that here is the start site. If this is the start site, from here the RNA has to form. So suppose the RNA started to form like this. Okay. Now this RNA which has been formed here has to move from outside from here. But this exit site here, this is my exit hole. This exit hole is particularly blocked by this sigma 70 factor. So even this core RNA polymerase cannot move forward, and this particular RNA so formed cannot exit from here. Now this process is referred to as the abortive initiation. This is called as abortive initiation. Now this abortive initiation occurs for approximately 30 to 40 times, and approximately 30 to 40 times this newly synthesized RNA tries to come outside. Then afterwards 30 to 40 times after doing this, there is one unknown factor. This unknown factor comes, and this unknown factor changes the conformation of sigma, the sigma 70, from the closed conformation into the open conformation. So sigma 70's conformation is changed from closed to open, and when it changes from the closed to open, now it will be looking like this. Suppose this is my here core RNA polymerase. Here is the exit side of core RNA polymerase. Now the sigma factor, earlier it was like this, but now it will become in a way like this, so that the RNA so formed, the RNA whatever it is being formed, it can now exit easily from here. So this is how the RNA will escape out. The newly formed RNA will be escaping out, and this process is referred to as promoter clearance. And after the promoter clearance has taken place, the DNA-dependent RNA polymerase will move forward to the coding region, and then the coding will be done and the whole of the elongation of RNA will be done. Finally, it has to come on to the termination region. Now, on the termination region, it can be through two kinds. One of them being the row-dependent termination, the another one being the row-independent termination. So one of the termination is row-dependent and the another termination is row-independent. In the row-dependent termination, a factor which is referred to as row factor is required here. Here the row factor is not required. Rather than row factor, one protein called NUS-A protein is required. NUS-A protein is required. So now RNA polymerase has reached its termination site, and now it has to exit. So how does it exit out? So see, for the row dependent termination, I'll explain you here only. So let's assume that this is our RNA polymerase, and this RNA polymerase has synthesized the whole mRNA, whatever, and this is the last part, this is the last part of mRNA which has to now exit out, right? So when this last part has to exit out, there is a particular site on this. This site is referred to as root sequence, RUT. So this particular site is what we call as root sequence. You people can note down this in your copies also. This is called as root sequence. Now what happens is that as this root sequence along with this mRNA tries to exit out, at this point of time, there is this protein which is called as a row protein, of which we were talking about. Now this row protein will come, and the row protein will bind to this root sequence. So here the root sequence and the row protein binding take place. Now as this has taken place and the row protein and the root sequence has bound, now what happens is that the mRNA so formed cannot move out. During this time, what happens is that this mRNA here, it undergoes capping, so that the exonuclease and the other important proteins do not attack this mRNA. And here there is a formation of a loop which is referred to as stem loop. The stem loop formation takes place. So this is my stem loop. Now as soon as the stem loop formation takes place, it is the characteristic feature. As this stem loop is formed, the row protein gets dissociated from the root sequence, and now this whole of the mRNA along with the stem loop gets free and it comes out. So in this way, the row dependent termination takes place. Now row independent termination is also very similar to the row dependent termination. Just remember that except for the root site which we had here in the row dependent termination, here we will have the U-rich sequence. Instead of root sequence, we have U-rich sequence. And instead of the rho protein, here we have the News A protein, and this News A protein will actually recognize this U-rich sequence and then will form the stem loop, and finally this RNA will be removed out from the this RNA will be removed out, and this particular polymerase will also be removed out, and this will be accompanied by a torpedoing movement. So this is how the whole of the process will take place. Also, there is another set of proteins which are referred to as GRE proteins. So these GRE proteins play also a very important role. GRE proteins will do the proofreading activity. So what they are going to do is they will try to have the proofreading activity on the incorrectly incorporated nucleotide in the mRNA. Now please remember that DNA-dependent RNA polymerase does not have any proofreading activity. Because of that, it cannot recognize the wrong nucleotide which has been incorporated. So in the place of that, there are these GRE proteins, and these GRE proteins will do the proofreading activity and then will remove those incorrectly inserted nucleotides into the mRNA. So this is how your whole of the transcription in the prokaryotes take place. In the next video, we shall learn about the transcriptions in the eukaryotes also.